and temperate forests all over the world. Just a sec. How these forests all over the world suffer from drought. So basically from too long or too intense periods of insufficient water. Many studies all over the world have demonstrated the impacts of water limitation on tree growth. And to some extent, these effects are similar between different species. So in this graph on the right, you see a study by Hoffman et al., who studied variation in growth for 3 different tree species, maple, cedar, and pine. And they measured the annual growth, which is reflected here on the y-axis, over consecutive years that are on the x-axis, using tree ring analyses. And the red vertical lines, I added those, indicate years with very low rainfall. For instance, 2003, as you can see here. And all trees of these 3 tree species showed much slower growth rates in those very dry years. So droughts seem to affect these different species quite similarly. But at the same time, drought effects can differ between species, and they depend on other resources that are available and that are needed by plants. So for example, here in this graph, you see the growth of beech trees plotted along a soil nutrient gradient here on the x-axis and a soil moisture gradient here on the y-axis. And the different colors refer to different growth levels. Red, very slow growth, and blue, very high growth. So as you can see, beech trees performed best or showed the highest growth rates on soils that were high in nutrients and that were moist. But here we see the same graphs for some other species. And I don't want to get into the details here, but what I want to point out is that while beech species, these ones, did best on moist and fertile soils, fir tree growth was actually kind of independent of water availability and mainly changed with changes in soil fertility, so along these x-axes. And oak trees over here grew best on soils that were high in nutrients, but relatively dry. So the amount of different resources affects tree performance, and these effects may differ across species. So I'm interested in how plants deal with such environmental change to grow and survive in a changing world. And my work mostly focuses on variation in soil resources. So to find out how plants deal with changes in water and nutrient availability, we need to look at their roots. But before I get into that, I want to explain you something about roots. When you think about roots, you may picture this. Some of you may also think about this, but I hope most of you, being ecologists, have this picture in mind. But the majority of the roots that we're seeing in this picture are what we call transport roots. Their main function is to transport resources between the crown and the soil, or the fine roots, and to anchor the plant firmly into the soil. And they are not involved in resource uptake. The roots that I'm focusing on are these ones, the tiny, most distal root tips that are the most active in resource acquisition. And as such, these are the ones that most strongly change in response to soil resource availability. So when I talk about root functional traits in this talk, I refer to the properties of these fine roots that we generally call absorptive roots, because their main function is to take up water and nutrients. So I use a trait-based approach, and I measure usually root functional traits that are root properties that are measurable and that determine the fitness of a plant. So survival, growth, and reproduction. And there are some that are more commonly measured or more frequently measured than others. And here's a short list of those. The meaning of most of them is pretty obvious, but I just want to explain a few here that are not always very easy to grasp, but that are very important to understand my talk. Starting with specific root length. So specific root length is basically the root length divided by the root dry mass. And in more functional terms, It's just the root uptake area per the amount of biomass that a plant invested in producing that root. And to get a better idea of what that looks like, if this differs, let's look at an example. So say we have two species and we give each of them 1.5 gram of biomass and we tell them to make the best root with this amount of biomass in their environment. So species 1 lives in this tough environment that's dry and it has very compact soils. So this guy aims to produce a root that is robust and can live long. So for this amount of biomass, this plant makes a thick root, but because he only has 1.5 grams, this root cannot be very long. So for him, the optimal root is this short, fat, stubby root. Species 2, on the other hand, lives in a very productive environment with lots of competition from other plants. So this guy wants to make a very efficient, long and thin root to quickly capture resources. So for the same amount of biomass, different species produce different roots depending on their environment, and this is reflected by their specific root length. So when you calculate specific root length here, you see that this species has a high specific root length, which contributes to uptake of resources, while species 1 has a 5 times lower specific root length and 2 times smaller uptake area, which contributes to the lifespan of roots. So it can retain its resources that are stored in the roots over the longer term. Then we have root diameter, which is a functional trait, and it's just the thickness of a root. And we have root tissue density, which is kind of the root analog of wood density, and it's calculated as the root mass divided by the root volume. So we have the two species again, and this time they have to make a root that they have to make roots of equal volume, so 4 cubic centimeters, and they can choose how much biomass they want to invest in it. So species 1 needs a strong root, so it invests a lot of biomass in this small root bit. And species 2 wants to save biomass to produce more roots, so it invests only a small amount in making this root. As a result, species 2 has a root that has a 5 times lower root tissue density, but it leads to higher uptake. Whereas species 1, again, produces this more robust root with longer lifespan. Another trait is root nitrogen content, and that is basically drives resource uptake because root nitrogen is assumed to be associated in root metabolism, and it increases the uptake capacity. And there's root carbon concentration, and this trait basically, a high root carbon content basically means that the root is protected, for example, from herbivory. Okay, so how plants deal with environmental change? The idea is that there's these two root trait strategies. So on the one end, we have traits that help the tree to acquire resources very quickly. And this acquisitive root trait, this acquisitive strategy is reflected by a root trait syndrome, where plants have roots with a high specific root length, a high root nitrogen concentration, and fast root growth to explore the soil very quickly. Then the other strategy is this conservation strategy. So these species produce thick and dense roots that are protected against herbivory by high carbon concentrations and that have long lifespan. This implies, so the acquisitive strategy implies fast water and nutrient uptake, which leads to fast plant growth on productive sites. And a conservative strategy means long-term retention of resources that are scarce, which leads to high plant survival rates in adverse environments. So together, this range along these two root trait strategies is what we call the root economic spectrum. So species can presumably be placed along the spectrum based on their root traits, like these guys over here. And this tells us then something about how they grow and how they survive in different environments. So this sounds like a very cool and convenient framework, but there's more and more studies that show that it doesn't really exist in the field. So studies show, for example, that root traits are not necessarily correlated in syndromes, and these syndromes do not necessarily trade off. There are, for example, many species that have both a high specific root length and they produce very dense roots. And then in the field, there's also no clear evidence of correlations between these two root trait strategies and plant growth and survival rates. We also tested this root economic spectrum in a review and meta-analysis, and we also found no evidence for it. And we came up with several reasons why a root economic spectrum does not exist. In the first place, roots work in a very complex environment. So for example, leaves are mostly optimized to capture light, which is more or less predictably distributed throughout the crown. So you have high light levels here and low ones here. So the leaves that are below can be optimized by having different traits than the leaves that are exposed to high light. But roots, on the other hand, need to not only acquire nitrogen, but also water, phosphorus, potassium, and about a dozen or so other micro nutrients. And all these resources differ in their availability, their mobility, and they can be quite unpredictably distributed throughout the soil matrix. And to complicate things more, these different resources may actually select for different root traits. So for example, nitrogen usually becomes available from mineralization of organic matter in the topsoil, and it's very mobile. So in a nitrogen limited system, trees may just enhance their root uptake area by increasing their specific root length in the topsoil, and they will get enough of the nitrogen. But phosphorus, on the other hand, is less mobile and it's more difficult to find. So in a system where phosphorus is limiting, roots may have to be very long and grow fast to find it at first. And once they reach a phosphorus-rich hotspot, they have to rapidly branch out, produce as much root tips as they can to capture as much of the phosphorus that they basically can in a short time frame before other species take it. So what's important here is that the limiting resource on a site defines what an acquisitive root looks like. And this is not necessarily a root with a high specific root length. And to deal with these highly heterogeneous soils, trees and plants rely on many other root trait strategies than those that are represented in the root economic spectrum. For example, they can produce these beautiful cluster roots in a very low phosphorus environment. They can vary their root hair length or their root hair density or their root branching or their rooting depth. And almost all plant species depend on mycorrhizal symbiosis to acquire nutrients. So all these different below ground strategies are available to plants, and they need to be considered too when we try to link root traits to plant growth and survival and environmental change. So to conclude this first part of my talk, we argued that there is not a single root economic spectrum, but instead that root traits are organized in a multidimensional trait space. And this multidimensional space needs to consider the complexity and heterogeneity of the soil and the multiple strategies and associated traits that trees can change to handle this variation. But which of these multiple below ground strategies and associated root traits really matter for growth? So in the second part of my talk, I'm going to focus on our test of one of these potentially relevant root traits, and that is root mass. So the root economic spectrum focuses on differences between species. But when we look at differences within species, we find that root mass is very often a major adaptation to resource availability in the soil. So for example, we tested differences in root traits between beech and spruce trees growing on a fertile and infertile forest soil in the Netherlands. So here you see the beech data, and here's the spruce data on the fertile and the infertile soil. And on the y-axis, you see the root biomass, basically. And we found that both species showed a very strong increase in their root mass on the poor soils. And this way, they could just enlarge their root surface area and capture more nutrients and water. And interestingly, they did not change their specific root length or their root tissue density. So there's definitely something to say for root mass and its importance in resource uptake, but it's only very rarely quantified across species in the context of resource economics. So we used a modeling approach to test if root mass is indeed a relevant dimension in a multidimensional trait space. And I'm going to walk you through the model. So we have a whole tree model that basically simulates the net carbon gain, which is our proxy of plant performance, as the carbon that is gained through photosynthesis minus the carbon that is lost through the turnover and respiration of tissue. So these three different carbon processes are basically calculated by multiplying the mass of a tissue times a certain rate. So for example, net carbon gain through photosynthesis is calculated by multiplying the leaf mass times a certain photosynthetic rate. So a key player in this above ground resource capture is leaf area index. And leaf area index is just the leaf area per unit of ground area. So it just tells you how much area does a tree have to intercept light and to fix carbon. So an increase in leaf area index leads to higher net carbon gain through photosynthesis. But having more leaves also means that more carbon is lost to the turnover of leaves and leaf respiration. In order to get a high leaf area index, plants and to obtain high photosynthetic rates, plants obviously need water and nutrients that are in the soil, and that's where the root traits come in. So in our model, we incorporated two root traits of interest. So we have root mass and specific root length. Remember the thick fat root and the fat short root and a long and thin root. And an increase in both traits leads to an increase in resource uptake and therefore leaf area index and net carbon gain, because both the root mass and specific root length lead to an increase in the root surface area. However, there's also some carbon cost involved. Because if you have a high root mass, it means that you also have carbon losses due to root turnover and due to root respiration. An increase in specific root length, however, comes for free. Because if you remember, plants can make more root surface area for the same amount of biomass. So an increase in specific root length leads to more water and nutrient uptake, but does not incur any additional carbon cost to the tree. And then we tested how does variation in these two root traits influences plant performance over here through these above below ground interactions. And this is our result. So on the x-axis, you see the variation in specific root length that we varied, and on the y-axis, the variation in root biomass. The different gray scales indicate the net carbon gain. So the darker the gray, the higher the net carbon gain, so the higher the performance. And what we see is that the highest net carbon gain was obtained over here. So the optimal performance of a tree was reached by having roots that have a high specific root length and by having low root mass. This way, sufficient resources could be acquired, but there was not a lot of cost involved because root biomass was low. Moreover, we saw that very low or very high root mass here in these white spaces led to tree mortality. Basically, in these white spaces, the carbon balance of a tree was negative. Apparently, either the carbon cost of a high root mass was too high, or there were not enough roots to capture enough resources. And now I usually look around the audience to see if there's any puzzled faces. But obviously I can't see it, so let me know if it's unclear. What I also want to point out is that a low specific root length and a high root biomass can still be adaptive, because trees over here still had a positive carbon balance. However, we need to revisit our model a little bit more, because we said that a high specific root length comes for free. but it doesn't really. Because as I said here, trees that have a low specific, roots that have a low specific root length generally live longer. So there is definitely a carbon cost involved in having high specific root length because these roots have faster turnover rates. So we added that to the model as well. So an increase in specific root length now means roots that turn over faster and therefore also presents a carbon cost to the tree. And then we ran the model again to see how this influenced tree performance. And here you see quite a similar figure, but with a very drastic difference. So now the optimal performance was over here, where trees had low specific root length and high root mass. So apparently those turnover costs are quite important for the tree's carbon balance. So what this work shows is that an increase in root biomass can be an alternative strategy to increase the root surface area and the carbon gain at the whole plant level, but only when you account for the carbon costs that are involved in having high specific root length. And then we wanted to see if this idea also held in the field. So we sampled the roots of 10 different species that are listed over here, again in the Netherlands. We determined their specific root length and root mass, and we plotted them in our simulated performance landscape. So each symbol here represents the specific root length and the root mass density of a given species. And we saw that across these 10 tree species, there's a trade-off between a high specific root length and a high root mass density, reflected by this negative relationship here. So for some species, root mass in the field indeed proves an adaptive strategy, the ones over here. So the second part of my talk, let's get back to the overall question. Plants deal with environmental change by more than just two below ground strategies. And we conclude that a root trait space is multidimensional. We also found that finding root mass can be an adaptive strategy and can therefore be an important dimension in this trait space. And currently there's a lot of other cool studies that looking at other strategies as well, such as mycorrhiza and fine root branching and fine root plasticity. But what our field data also showed that which strategy is best might depend on the species. And that's what the final part of my talk is about. So here in this study, we looked at how plants deal with environmental change, but within species rather than across species. So basically, do these two strategies of resource acquisition and resource conservation and their associated traits, specific root length, root density, root diameter, occur within species along an environmental gradient? And we addressed this question in the beautiful French Alps along an elevation gradient. So we had this elevation gradient from 1400 to 2400 meters above sea level. And at the lower altitudes, the soils were more fertile. And at higher altitudes, temperatures were colder and rainfall was higher. We then set up 5 replicate plots at each altitude, so with an increase of 100 meters in altitude. So basically 5 replicate plots at 1400 meters, 5 replicate plots on 1500 meters, etc. And in each of these plots, we sampled plants, one plant for each of 11 different species. And these different species included grasses, forbs, shrubs, and trees. So based on what I've explained before, we could hypothesize that with increasing altitude, the environment becomes more harsh. So this could lead to more conservative traits. So we expect that within a species, plants increase their root diameter or their root tissue density to handle this adverse environment. But a high specific root length can also be beneficial because these long and thin roots are very efficient in searching the soil and exploiting its resources. So alternatively, some species may also opt for producing more acquisitive roots on high altitudes. But these are our results. So here we have 3 graphs, each of them representing 3 different traits which we expected to increase for at least some species. So here we have specific root length, tissue density, and diameter. On the x-axis, you see the elevation gradient, and the lines represent significant relationships between a trait and elevation per species. So each line corresponds to a different species. And I only plotted the lines for significant responses. So I don't want to get into too much of the detail because we don't have time. But basically what we found here is that only one species increased its specific root length. We also expected that some might increase their tissue density to produce sturdier roots, but only 5 out of the 11 species did that. And increasing root diameter was only observed for one species. And basically, all the other species showed very different responses. So they showed linear decreases, linear increases, bell or U-shaped responses, or no response at all in these 3 root traits along this environmental gradient. So basically, within species, root trait variation along this environmental gradient was very idiosyncratic. And this may be partly because of the multivariate nature of our gradients and from species differences in how sensitive they are to their environment. So for example, with increasing altitude, rainfall increased and temperature decreased. So for trees that grow here and towards the tree line, it means that they face a shift from water to temperature limitations as we go up the mountain. But grasses at low altitudes, they grow in the forest and they may be outshaded by trees and light limited at low altitudes. But when you go up the mountain, they may be nutrient limited. So this implies that different species respond to very different environmental limitations along what we think is this one common environmental gradient. And in turn, these different gradients may call for different trait adjustments. It's like I said before, the environment determines what an acquisitive root looks like. So for example, light limitations for the grasses here might trigger a decrease in tissue density because this requires to invest more carbon in your root, while nutrient limitations may stimulate mycorrhization. So different limiting factors along the gradient may trigger different trait responses across species, resulting in idiosyncratic patterns. And then there's also traits that we did not measure. So plants may increase their root to shoot ratio or their root biomass when we get up the mountain to deal with low fertility there. And these responses may be more pronounced at the whole plant level. So drought may stimulate plants to increase their specific root length, but they can also reduce their specific leaf area or their stomatal conductance. So quite likely, how plants deal with environmental change is reflected by a combination of simultaneous above and below ground trait responses. And this is something that I'm looking into in my current postdoc project. So we are looking to explain tree growth by combinations of leaf and root traits, soil data, and tree mycorrhizal association. And we do this using the censuses collected at the Big Woods plot here in Michigan at the George Reserve, and using trait data that Natalia and her co-workers collected last year for leaves and roots. And we're also planning to do this across northeastern US forests using existing data set. And I'm doing this work together with Natalia Almagna here at University of Michigan and Dr. Jenny Zambrano at Washington State University. And then I want to share this picture, this beautiful picture that we took this summer at the biostation with some grad students, Natalia and I. And then to finally really wrap it up, how do plants deal with environmental change? Well, they do so in many different ways that may differ between species and that likely depend on the environment. This implies that we cannot explain trait performance relationships using a root economic spectrum, but we need a multidimensional conceptual framework, at least below ground. And this should include environmental variation, these multitude of strategies, including root biomass, and it implies that we need to measure more other and newer traits rather than the, 5 or 6 that I listed at the beginning of this talk. And finally, my model showed that a whole plant approach can help explaining environmental effects on plants, because plants respond above and below ground. And these responses also interact with each other. And that brings me to the end of my talk, where I want to acknowledge all the help I got washing roots, and digging up roots, and scanning roots at Wageningen University, with my coworkers in Montpellier, France, and here at the University of Michigan, and collaborating with Jenny Zambrano. And if you think, wow, this is really cool stuff, I should also measure roots, I want to highlight a paper that we recently published in an online archive, which is a starting guide to root ecology. It's, I don't know if you know it, many of you might know the Cornelissen Trait Handbook. This is a similar handbook, but then only for roots. And it tells you everything about how you measure roots, what root traits mean, how you wash them, basically everything there is to know. It's open access, and you can download it from the link here, or hopefully using this QR code that I made. I don't know if it works. So please have a look. And finally, thanks for your attention. If you've made it this long, I'm happy to take any of your questions. Wow, this is really an uncomfortable silence.
I wanted to say, so if you want to either put your question, write it out in the chat, or you can also just ask to speak verbally if you want to ask your question too.
Hey Monique, this is Camille talking.
Hey.
So that was an incredibly really good talk. Thank you so much. I feel that I learned a lot, even if I'm working on roots, so that's great.
Wow.
So at the end of your talk, you mentioned that you are now exploring, the whole, taking the whole plant perspective approach and looking at the different mycorrhizal types. So are you gonna look at arbuscular and ectomycorrhizae, and then look at the traits that you've already measured, and then are you gonna look at new traits too?
Sorry, am I gonna look at, you mean, you probably are saying mycorrhizal traits?
Yeah, mycorrhizal traits, and then new root traits too.
Yeah.
So physiology or anatomy, I don't know.
Yeah, no, so I hate to disappoint you, but we're not looking at mycorrhizal traits in this particular study. Although I really do think that would be, like a super important part of this whole plant approach. It should be like the whole plant and associated fungal approach, because, well, I don't have to convince you, but fungal traits also matter. They also present a carbon cost to the tree. They provide a lot of nutrients and water to the tree. So they would definitely, it would definitely be possible to integrate them into our model conceptually. We do need more data on that, but that's something I really think that's one of the ways to move forward. I hope that answers your question.
Yeah, it does. So, thank you.
Yeah. Oh, and I should say, so we only just, so we collected some root traits and leaf traits on these different tree species. And we just separated them as ecto or arbuscular mycorrhizal species. So we did not measure, for example, the extent of pollinization or so. It was just mycorrhizal type as the main trait.
Monique, I have a follow-up question, because I feel that every time, we look at these big climate gradients and we look at intraspecific or interspecific variability, and it looks like there are too many things that varies and species are different. So if you were to design a study to investigate linkages between the traits and the environment, would you use a climate or environmental gradient again, or would you do it in the lab? What would you do?
Yeah, that's an interesting question. And I'm actually, I'm thinking about my future research. I'm, working on drafting my research statement. And I think the answer lies in combining them. So I really think before we can scale up, we need to scale down. So I think we need to find out which are the traits that really measure in different environments. And I think we need to do this using an experimental approach under quite controlled environments, because as you said, everything changes at the same time. And by identifying those, I think we can use that information to figure out what kind of environmental gradients we should be looking at in the field. Although I'm the first to acknowledge that we cannot necessarily one-on-one translate experimental results to the field, but I think that will be, I think we really need to do both. I think we should look at the smaller scales and at the larger scales at the same time, and have this crosstalk between the different scales. You know, look at experimental data first at the smallest scale, maybe single root or mycorrhizal traits. Then look at how it changes across the environment and see if those results don't add up. We go back to the lab again and test new hypotheses.
Looks like you have a question from Luis. He's asking, how plastic are these root traits? If you watched a single tree over time, how much would they change? Trying to wrap my head around trees. They're so much bigger than microbes.
Yeah, they are. Yeah, that's a good question. So when I look at plasticity or intraspecific trait variation, I compare the same plants at one moment in time, but across different environments. But I think there's also ontogenetic effects on traits. So definitely from seedlings to mature large trees in a forest. I think root traits would definitely differ. And I think that also makes it hard to, extrapolate seedling data that are easier collected in the labs to the forest and to larger trees. So I think there's quite some plasticity, but I don't really know how it works along the ontogeny of trees.
Okay, and now a question from Juan asking or saying, thank you so much for an awesome talk. I'm curious about how soil type may influence root traits.
Yeah, so the short answer is a lot. The longer answer is that it really depends on what variable you're looking at. So I think, as I also tried to point out in my talk, if you look, for instance, we shouldn't treat soil as one, box that influences root traits. I mean, there's the water, there's the nutrients, there's the different nutrients, and as I said, all of them influence traits in different ways. So, yeah, I think it's gonna depend on what kind of soil environment you're looking at, but the soil in itself has a huge importance on roots. I mean, roots go where they can go, so physical and chemical properties determine root traits, and they go where they should go, so where the food and the water is. I hope that answers your question. There's really a lot of studies on this, so.
Do we have any more questions? You have quite a few people who thanked you for a great talk, Monique.
That's good. That's good. I'm glad.
Oh, we have another one from Jim. Sunlight is very directional. Shading is important. What is known about the three-dimensional direction from which resources come?
Ooh, well, that's more a question for a soil ecologist, I suppose. But it's not as directional as light. So for resources that mainly come from decomposition of organic matter, it might be, there are usually higher nutrient levels at the topsoil than at lower soil, at deeper soil layers. For water, it might be the same, but in one of my systems, it's in the Netherlands, so almost underwater. So the groundwater is about 50 centimeters, 100 centimeters. So the availability of water is much higher at deeper soil layers, depending on where the groundwater level is. And then there's like rock-derived nutrients like phosphorus, which becomes available through weathering, besides from other sources. So yeah, that may be a bit less predictable. So definitely multi-directional, depending on the resource though.
Okay, great. Now Hector is asking, was curious about the degree of phylogenetic conservatism in root traits.
Yeah, that's something that there's some really cool studies about them, and most of them are first authored or co-authored by Oscar Valverde. If you're really interested, you should definitely just Google him and read his papers. But there's definitely there's definitely strong conservatism, phylogenetic conservatism in root traits. I cannot say at the moment exactly which traits are more strongly conserved than others. I didn't take it into account in my studies, but if you look, for example, at global scale studies, they always correct for phylogeny. So there it really plays an influence. And there's definitely trait differences or even mycorrhizal differences that disappear when you take phylogeny into account. So it's definitely an important factor.
Can I ask a live question?
Yes, please.
I'm trying to wrap my head around organs that have to do lots of different things all at the same time. So, you know, in thinking about an organ like a lung, it's only got two things to do, right? It's got to take oxygen in and expel CO2 out. And so I can imagine that's a trait where you might be able to measure something relatively easily, whereas a root, it's got water, phosphorus, iron, calcium, all the things that you mentioned, never mind a myriad of micronutrients that we all ignore. It seems to me that it might be extremely difficult then to tie any particular root morphology to all of those different functions. And perhaps enzymatic activity might be a better suite of traits to use rather than the morphology of the roots. In other words, the functional aspect of the root is not how fat it is or how thin it is or how long it is, but what enzymes are active in the root. Would that be an alternative way to approach it?
I honestly don't know too much about these enzyme activities. I know that they're being applied for mycorrhizal studies, for example. So yeah, that would be something to look into. I think there's also studies that are doing that. Maybe not directly their enzyme capacity, but for example, looking at what kind of chemical compounds that roots exude that influence the microbiome and the availability of resources. So yeah, I think it will be interesting to look at. I don't know how feasible it is to do it in the field, especially in a mixed forest with lots of different species and understory vegetation.
Well, no harder than finding the roots. I guess what I'm suggesting is I think you're measuring the wrong traits.
Oh yeah, I think so too.
The traits of interest are the traits involved with the acquisition, which is actually the enzyme activity and not necessarily the shape of the root.
Well, so I don't know about the enzyme activity. I don't know if there's any data on that and if that's been linked to resource acquisition, but I definitely think we're not measuring the right traits. Yeah. I think that, and the thing is, most of these relationship between a trait, especially morphological trait, and a function are based on what we observe in leaves. So for example, specific root length is supposed to be this super important trait, because specific leaf area is so important above ground. And I think we should really abandon that whole idea and look at different traits. Yes.
Great, thank you.
You're welcome. Great questions.
I guess if that's it for questions, we can all give Monique a round of applause silently, I guess, if you're. But thank you so much for giving our first talk of the semester, and join us next week. We have another speaker on Thursday, October 1st at 3 p.m. Thank you all.
Thank you for inviting me and for coming to watch.
Yeah.